Hi friends, this video is about the viral replication. I have done a video about what is virus, what are the classifications of virus, and what is structure of virus. If you haven't watched these videos, please do watch it before you go to the viral replication. So what is a viral replication? Viral replication simply means that it is the replication of virus, and this replication only happens when it is inside a living host cell. So when it is inside a living host cell, the viral replication takes place and it synthesizes new viral particles. So these new viral particles are later assembled so that a correct structure of virus is formed, and these are called as virions. So later these virions escape from the cell to infect the other cells. So a viral replication only happens when it is inside a host, and new viral particles are synthesized when it is inside. These synthesized particles are assembled so that a correct structure is gained. So these correct structure viruses are known as virions, that is, the infectious particles of virus is called virions. These virions later escape and infect other cells. Now let us discuss the first step of replication. The first step of replication is attachment. So in this step, the virus goes and attaches to the surface of the host. So in this attachment, there is a specificity existing. The envelope spikes get united with the surface receptors, that is, the receptors which are present in the pili or flagella of a bacteria or which are present on the host cell membrane. So the spikes get united with the surface receptors of the cell, and hence the attachment is done. So the next step in viral replication is the penetration. Penetration is the step in which the viral genome enters into a cell. So in this step, this may occur either in any of the three steps. First is phagocytosis, and second is a method in which the envelope of a virus may blend with the cell membrane. Third method is which the virus may inject its genome into a host cell. The last method, that is, a virus injecting its genome into a host, is seen in bacteriophages. The tail of the bacteriophages attaches to the cell membrane, and the enzymes open a hole in the wall. This leads to the penetration of the DNA into the hole, and the DNA reaches the cell. The next step is known as uncoating, and in this step the capsid is being stripped away from the genome, that is, now here the genome is free, and this free genome, if it is RNA, it acts as a messenger RNA, and it provides genetic codes for the synthesis of enzymes. So the third step of viral replication is the uncoating step. In this step, the protein coat is being stripped away from the genome, and the naked genome is present in this stage. The actual replication process starts from this stage. These enzymes are used for the synthesis of viral genomes and even the capsomeres, that is, the subunits of capsid. These then arrange and assemble and forms the components of new virus. So in this step, if it is DNA is the genetic material, then the synthesis of messenger RNA molecules proceeds, and the process proceeds. So till now the virus has come and attached to the cell, and this is known as adsorption, after which they are attached, the penetration occurs. After the penetration, the replication, that is the actual replication, happens. After this stage, the assembly occurs, and after this matures, that is the virions matures, and after it is matured, they are then released. That is the last stage of viral replication, is the release of virus. So this can be done in a number of processes. So for example, the host cell may be biochemically exhausted and it may be disintegrated, and thereby this releases the virions. So another method in which the enveloped viruses are released is by budding. So in this process, the nucleocapsids move towards the membrane of the host cell and they force themselves together, that the membrane is in a process called budding. So during a budding, a portion of the cell membrane pitches off and surrounds the nucleocapsids as an envelope. Another way in which the release occurs is by lytic cycle. So in this cycle, the viruses are free to infect and replicate in other host cells in the area. In this method, the host cell experiences death by a lytic cycle. So not all the viruses undergo lytic cycle reproduction. In some viruses, they become active in the host cell for a long period and they do not replicate until then. So this cycle is called lysogenic cycle. So these viruses are called template viruses or proviruses, and this is because they do not cause the death of the host cell immediately. The different ways in which a virus is released from a cell is by: first, biochemically by exhausting; second is by budding; third is by lytic; and fourth is by lysogenic cycle. So these are the ways in which a virus is being released. So now let us recall the 6 stages of a viral replication. So they are: first is adsorption, second is penetration, third replication, fourth is assembly, fifth is maturation, and sixth and the last step is release. This diagram explains the overall viral replication. First step, which is the attachment of virus to a host cell. So after this virus gets attached to the host cell, it penetrates inside. And after which they are penetrated, the uncoating step occurs. That is, the protein coat is removed and the naked nucleic acid enters. So the synthesis of nucleic acid and proteins occurs. The next step is the assembly. They assemble so that the virus are formed, that is, the virions are formed. Next stage is the maturing of the virus, after which mature viruses are obtained, they are released. So this is the steps involved in a viral replication. Hope you understood the concept of viral replication. For more such videos, do subscribe to our channel. If you like the video, please like and share with your friends.